Okay. So, it's been a while. So, does everyone remember what class this is? Who enjoyed a little bit of a break? Should I do that more often? I don't know if anyone else would appreciate that. But so I intended on recording this lecture in advance and posting it, but YouTube was acting funny and wouldn't upload anything, and I just finally threw my hands up in there and said, well, they don't want to watch it anyways, and we'll just wait. So I was trying to be an overachiever, but okay. So where were we? We were talking. Oh, right, business. So obviously no recitation this week. Anyone who thinks there's recitation today, you are wrong. Most of you gave me your Scantrons for the exams. There's a handful of you who need to still get them and give them to me. Bring them next week for recitation, and there will be a quiz this weekend on this stuff for next week. Okay, so what have we been doing? We were talking about protein import into the ER as we start talking about sending proteins to where they need to go to do what they need to do, and we've started with the secretory pathway. We talked about signal sequences in terms of the sequence that's recognized by the SRP that brings the protein, the newly or early translated protein to the ER, and the steps for import or export, depending on your philosophy, to the ER. We spoke about integral membrane proteins and how membrane proteins are put into the ER membrane because that ER membrane will eventually become the plasma membrane. And then we started to talk about what happens in the ER. Lots of enzymes that help in folding mostly. And so now we're ready to leave the ER, well, pretty much ready to leave the ER and go to the Golgi. Is everyone up to speed? You all remember learning all this? Everyone's ready for the test? Okay, good. Okay, but before we can leave the ER, we need to be folded properly, because what happens if we are not folded properly and we leave the ER? Is that good for the cell? No, because misfolded proteins aggregate, cause trouble, don't function. So there needs to be quality control. And so ERAD, ER-associated degradation, is one mechanism for, major mechanism for quality control. And it doesn't let proteins that are not folded properly out of the ER. And most people really understood this on the quiz, so that was good. And then recitation next week is going to be a paper about this. A lot of these figures are from that paper, so it should make reading the paper a bit easier too. So basically, the way it all works, this is just a summary, is, so here's your ER, and you're going to have import if there's your ribosome and your RNA, and you've got your import of your protein into the ER, and this huge sugar is added to your protein. Every protein that comes in, big block of sugars added to it, and what happens is there's this trimming of sugars and adding of sugars that acts as a signal to let basically to let the cell know whether or not the protein needs to be retained in the ER or whether or not it could be shipped out of the ER. And if it's not folded properly, then the sugars bind to calnexin and calreticulin, and those are shown here. Calnexin is integral membrane protein, so it's actually in the ER membrane. Makes sense, right? If you want to retain something to the ER, kind of have something bind to it that's stuck in the ER. And then there's calnexin and then calreticulin, and there's this constant trimming and adding of sugars as the protein folds, and eventually it's folded properly and it leaves the ER in a vesicle. It buds out in a vesicle, goes to the Golgi. If it's interacting with these lectins or chaperones too long, then the ER goes, oh, this protein's having a hard time folding. Why might a protein have a hard time folding? What could be wrong with it that might result in it folding wrong? Right? Because we talked about protein folding, and how usually it's just based on the sequence in a way, right? The sequence, all these amino acids have chemical properties, and they're going to want to interact in a certain way. Yeah.
The gene it came from could have mutated.
Right. So that's exactly what I was looking for. The gene it came from could have been mutated. So if you have a mutation that changes an amino acid, then that could result in it not folding properly, right? Because remember, it's those biochemical properties of amino acids that determine the structure, okay? And so if suddenly, let's say there used to be a cysteine, which is a nice small amino acid, and there's some mutation that changed it to a phenylalanine, which is gigantic. It has this huge ring structure. And let's say that cysteine used to be buried in this tiny little space, and suddenly you've got this gigantic, huge golf ball thrown in. Is that going to allow the protein to fold in the right way? No, right? It's going to completely be wrong and not stable and probably be retained in the ER. And so when it's retained, so when it interacts for too long with these chaperones, it'll be retrotranslocated. So that's basically kicked out of the ER through a translocon, and it'll be ubiquitinated and degraded via the proteasome. So that's the summary of how it all works. And the basics are just that doesn't fold right, gets stuck in the ER, stays in the ER too long, gets kicked out and degraded. And it's all determined by the sugars. And so what do I mean by sugars? So here's a summary of the sugars. So this here, and this is straight from the paper that you guys are going to read for recitation next week. This is the block of sugars that's added. And basically how it works is that you'll get 2 glucoses. So the yellow triangles are glucose. 2 glucoses are removed by glucosidase. What's a glucosidase? An enzyme, right? ACE, glucosidase, right? Okay. Just making sure. So glucosidase is going to remove these 2 sugars here, and there'll only be 1 glucose here, and that's what's recognized by calnexin and calreticulin. And it retains the protein in the ER, facilitates folding, and then when the final glucose, this triangle, is removed by another glucosidase, it's released, it can go out, it buds into Golgi and goes to where it needs to go. That's proper folding. Unfolded proteins are reglucosylated by an enzyme and called UGGT, and you put the glucoses back on, and what happens, it will associate with calnexin, calreticulin, it will fold some more. If it folds properly, it'll be chopped off. That last one will be chopped off and it can go on its way. If not, it'll be... So it's like that over and over and over again. This cycle is a folding cycle. And so then there's another enzyme, mannose, that removes, let's see, the green circles are mannose, another sugar. And so there's another enzyme that removes one of the mannoses, and that is kind of another thing that needs to happen to be kicked out. So if you are interacting too long with calnexin and calreticulin, eventually the mannosidase will come along, remove that mannose, and that will be the signal for kicking the protein out for degradation. Any questions about that process? It's actually pretty simple. It seems like a lot because it's a busy summary slide, but just go through it, and it's really all about, like, is it binding to something that's going to keep it in the ER or not? And that's all based on the sugars. So why is this so important? Why do we care about this? Well, just some examples of diseases that are associated with ERAD. Cystic fibrosis, right? That's a significant disease, right? The protein that's mutated is called CFTR, cystic fibrosis transmembrane conductance regulator, and it doesn't fold properly because of the mutation, and it's targeted to the proteasome because of through ERAD. And so that's funny. It's one amino acid. That's it. 1 amino acid. And it's like there's actually nothing wrong with the function of the protein. It's just that it doesn't fold well enough to get out of the ER. So if you could figure out a way to kind of stabilize it just a little bit to get it out of the ER. Well, there's your cure for cystic fibrosis. Juvenile Parkinson's disease, there is inefficient folding of a GPCR, a G protein coupled receptor, which we will learn about in gory detail. And it leads to ER retention, aggregation, and then it actually doesn't trigger ERAD. It's retained for too long, aggregates, and triggers the unfolded protein response. And then apoptosis. And then CMV, cytomegalovirus. So viruses are always trying to evade the immune response. So a smart thing that it does is it promotes degradation of MHC class 1 heavy chain. Does anyone know what MHC class 1 is? Anyone taking immunology? So it's part of the immune response. It's on your antigen presenting cells and interacts with your T cell receptors, and is very important in presentation of viral antigens. So if you're infected by a virus, then your cells will become decorated with viral antigens to let your body know that you are infected by a virus. And this virus is brilliant. MHC class 1, it's a membrane protein. That means it needs to go ER Golgi out to the plasma membrane. It actually promotes degradation of MHC class 1 heavy chain, so one of the chains of the protein, through ERAD. Brilliant, right? This protein's absolutely fine, but I'm going to grab it and degrade it anyways. Brilliant. Okay, questions? Good. No questions. Everyone loves it? Is everyone happy to be back with me? Okay. Good. Who wishes they were on the airplane with me the other day while my son cried for 1,100 miles? No. I wish someone was there to hold him. Then he fell asleep on the floor literally 10 minutes before we landed. And then there was like us in a row and another row with another 2-year-old who screamed also. And these elderly people in between, they were like the sandwich, like the meat in the sandwich of screaming child. Oh my God, it was horrible. I just kept saying, well, at least we're not going to Japan, because I did that and that was really bad. Okay, well, Japan was good. Anyways.
What does CMV stand for?
Cytomegalovirus. It's a herpes virus. Who here has CMV? I think about 40-50% of you should be raising your hands. Yeah, check it out. Most of you have CMV, and most of you, for real, most of you have CMV. And I'm not saying you have herpes. Well, first of all, anyone who's had a cold sore has herpes, so just get over it. But CMV, it doesn't bother you at all unless you're immunocompromised or pregnant and you get infected. Then it's really bad for babies. And then most of you also have Epstein-Barr virus, right? You guys know that. Who has Epstein-Barr virus? Who's had mono? Yeah, if you've had mono, you have Epstein-Barr virus. Anyways, okay. Any other questions? Good. Okay, so we're properly folded. We're let out of the ER in the correct way, not through the bad way that leads to degradation. I guess you could say it's a good way because you really don't want those proteins getting out anyways. And so now we're on our way to the Golgi. So proteins are going to move between compartments in the secretory pathway by means of vesicular transport. Transport in vesicles. So the proteins literally bud into these vesicles. So if it's a protein that's going to be secreted, it's going to be these little circles in the middle of the vesicle. And if it's going to be a membrane, or if it's going to be secreted or going to the lysosome, it'll be in circles. If it is a membrane protein, it's going to be in the membrane, and it buds, and it goes, and it fuses with the Golgi. And then things happen in the Golgi, and then it'll go to the next place the same way, vesicular transport. So just to remind you about the secretory and endocytic pathways. I always feel like I'm on display in this classroom. If I have the door open, people walk by and you're like, right there. Okay. Right, so remember, secretory and endocytic pathways from the ER to the Golgi, and then you'll go to secretory vesicles. So outside of the cell, you're going to be a secreted protein. You go to the endosome and lysosome. If you're lysosomal protein, you'll go to the plasma membrane. If you're membrane protein, right? And this is how it looks. Here's your ER, which is, remember, it's attached to your nuclear envelope. And there's your Golgi, and you see there's these vesicles that are kind of popping around, and they go to wherever they need to go. Okay, everyone happy? Yes. Okay, so transport vesicles have different coats with different functions, right? So you guys read all about this, clathrin, COP1, COP2. These are these protein coats that are involved in what? What do they do? What's their point? Yes.
I think they shape the vesicle.
Right, so they shape the vesicle. What else do they do? Come on, you guys answered questions about this. Yes.
It helps tell it where it's supposed to go.
It helps with telling it. It helps with telling it where it's supposed to go. Yep. Where else? Where else? What else? One more big thing. Yep.
They act as chaperones to keep the proteins from.
Act as chaperones? No, but you're kind of like, I see where you're going. They do something with the proteins inside the vesicles, or inside the ER, I should say. How do the proteins that are in, let's say, okay, so you've got the ER. It's this big thing, right? Where's my laser? There it is. This huge ER. Well, it's not that big there, but let's just say it is. And you're having not one protein being made, but many proteins being made. Some need to go to the plasma membrane. Some need to be secreted. Some need to go who knows where. So how do the proteins even get into a vesicle? Do they just randomly get into a vesicle? Does it have anything to do with the protein coat? I see people nodding. Okay, just remember that. We'll learn about it. Right, so it's involved in budding and literally causing that shape of the vesicle. And it's involved in binding to the cargo for the vesicle. And then as soon as it buds, the coat disassembles and it's there to be recycled for something else. So proteins, they leave the ER and COP2 coated vesicles. The function, like we just said, it concentrates certain membrane proteins in a region that will give rise to a vesicle, and it's involved in assembly into the curved basket-type lattice that promotes the shape of a vesicle. So here's how it works, right? So you've got your ER, and you've got these cargo receptors and cargo proteins, and what happens is you've got the different components of the coat that come and actually bind to the cargo receptor and help with packing the cargo into the vesicle and then helping it to bud and turn into a vesicle. So here's another picture. So here's clathrin, and this is a nice example of this. I always like it because it's very simplistic. Clathrin will bind to another protein called adaptin, which will bind to the cargo receptor. So here's your cargo receptor, which is in the membrane, and it will bind to the cargo. Let's say it's some protein that's about to be secreted, and then the clathrin will adaptin will bind to adaptin. All adaptin does is it helps clathrin to bind to the cargo receptor. And then you've got adaptin binding, and you start, as you have that curved protein binding, you start to get the curving of your membrane and formation of that budding vesicle. And then you've got the vesicle, and then Dynamin comes and tightens around the membrane and helps it to pinch off. And then it goes on its way. As soon as it pinches off, the coat disassembles and is available to make another vesicle. Any questions about this? I feel like I got a question from someone about the quiz about this. No? Everyone's clear. Okay. Okay, so Dynamin mediates the pinching off. So this is what it was. I had the question about if there's a deletion of some sort in Dynamin, what will happen to your vesicles, right? No Dynamin, no pinching off. So you'll have all these vesicles will be tethered, kind of like in this picture here. And then you won't have transport at all of your cargo to wherever it needs to be going. Okay, so Rab proteins. Okay, so now you're thinking to yourself, okay, that's great. You're telling me cargo gets loaded up into some vesicle, and then what? How does it get to where it needs to go? It just floats around and randomly binds to things and fuses with things. Well, that would be a horrible thing, right? Because it would start just binding and fusing to the wrong places, right? There's kind of an order, and proteins need to go to certain places. So Rab proteins are involved with this process. They're monomeric GTPases, so that means they're proteins that use GTP to function, and they determine the specificity of vesicle targeting. So when you think about GTP, I want you to think about something that's either on, GTPases. I want you to think of something that's on or off. You can think of it as a molecular switch, and we'll talk more about this. When it's GGP bound, it's inactive, and when it's GTP bound, it's active. And so it needs other proteins to help to get rid of the GDP and put a new GTP when it needs to be turned on. So just something to keep in mind. But so Rab, all it really does is it actually it interacts with membrane. So, but in this case, Rab GTP, it's active. So a Rab GEF, that's a guanine exchange factor that adds the new GTP. It'll activate Rab, and it will bind to a membrane. By membrane, I mean membranes of different origins. There are many different Rab proteins that might bind to many different membranes. I'm not talking about outside the cell or the plasma membrane in this situation. I'm talking here about ER membrane, Golgi membrane, lysosomal membrane, plasma membrane, but the inside, right? So all it really does is it acts to tether the vesicle to this Rab effector, which is just a tethering protein. In the book, they say it's like a fishing line. So I guess you can think of it like that. And it binds to this tethering protein, and the whole purpose of that is just to keep it, to bring it in close to the membrane of where it needs to fuse. And then it will interact with the snares, which are right here, and the snares help to fuse it to the membrane. So you've got tethering, docking, and fusion. Those are the steps. You don't even need to really think too much about it, right? Because obviously, if a vesicle needs to get somewhere, it makes sense that it would need to bind to something that's going to bring it close to the membrane it's going to fuse with, right? So just think of it as, we'll use the fishing analogy, that this Rab effector protein is kind of this fishing line hanging out there, and the Rab GTP will become activated, and it's like the fish, and the fish will bind the fishing. It'll bite the worm or whatever you're using to catch your fish. What do you guys like to use to catch your fish? Do you like the real worms or the fake ones? I grew up on the fake ones. Fake ones, real ones? Real ones are better. Who uses chicken wings? I'm just saying. I know chicken wings are the best way for crabbing. Who believes that? Okay, good. Okay. I know fish, different crabbing. I know. I'm from Florida. What do I know? Okay. So then the fishing line catches the fish, and you reel it in. He brings it closer, and then the snares catch it, and they twist up together, bring it really close, exclude all the water, and you get membrane fusion. Okay. You can also think of it as like, did you guys have those Velcro balls when you were a kid that you threw against the thing? So it's like kind of like throwing the Velcro ball against the wall. Anyways, so you've got so the steps are, you've got your Rab GDP, which is just hanging out, so it's inactive, and then this Rab GEF is actually on the membrane, binds to Rab GDP, exchanges it, becomes Rab GTP, so it's active. Then it goes and interacts with this effector protein, which brings it close to the snares, and then you have fusion. When you have fusion, what's the whole point? Release of the cargo into its destination, which in this place will be ER to Golgi. Questions? Comments? Recipes? I have a great macaroni and cheese recipe. Sorry, thinking about lunch already. Okay, different Rab proteins on different compartments of the secretory pathway, right? So you're saying to me, okay, that's great that you've got this thing about this fishing line and fish and fake worms, but how is this? What does this have to do with specificity? Because I'm telling you, Rab is one of the major factors. The Rabs and the snares are your major factors in specificity. Because the point is, you want to get your cargo from your ER to your Golgi. You do not want your cargo to go from your ER to your straight to your membrane, right? Or you want it to go from your Golgi straight to the membrane, or from your Golgi straight to the lysosome. You don't want it to go to the wrong place. So these Rab proteins, they actually bind to specific membranes. So there's Rab 1, 2, 3. There's a lot of them, and they're specific to different organelles. Actually, that'd be a really nice way. Let's say you just wanted to find you were doing some sort of experiment, and you were wondering if your protein went to early endosomes or late endosomes, right? You could actually do an experiment where you put your cells on a slide and do immunofluorescence, so microscopy using an antibody that's fluorescent, against a certain Rab protein, right? So is your protein in your early endosomes or your late endosomes? How would you differentiate? What antibody would you buy to detect your early endosomes? I hear someone saying 5C, right? 5C or 8. And your late endosomes, 7. So this is a really useful thing to do if you're studying protein localization. You can do it with immunofluorescence. You can do it just with centrifugation. Remember we learned about the microsomes and how you can disrupt your cell and get your microsomes, and then you put it in your centrifuge and you spin it down and it separates by density, and then you can take out 1 ml at a time into little tubes, run it on a gel, and you can actually see what's in each fraction, right? If something's in the same fraction, so let's say you think your protein's in the late endosome. Well, you can run a Western blot, use those same antibodies against Rab7, let's say, and your protein, and if it's in the same fraction, it was probably in the same place, right? Same density, interacting together. Anyways, this is very useful. Okay, questions? Okay, moving along. Okay, so more about Rab. So Rab, it's actually cooperative how Rab works. Rab activates more Rab, and you get these patches of Rab and Rab effector proteins on membranes. Just in a little bit more of a specific way, you've got your Rab GDP. It's kind of hanging out there, not active, right? GDP bound. And then your Rab GEF will come along and bind and activate, right? So now you got activation. And so it all actually has to do with this helix that's hydrophobic. And it's kind of buried when it's GDP bound, but then you have this conformational change when you get the exchange for GTP. Frequently, when you're dealing with GDP and GTP, you're dealing with changes in the structure, conformational changes. And this helix with a lipid attached to it unfolds. And when you have a lipid and you're near a membrane, an obvious thing to do if you're a lipid near a membrane would be to, what? Avoid the membrane or go near the membrane? Go near it. Hydrophobic loves hydrophobic, doesn't want to be near all that water in the cytoplasm. So it's going to go and bind to the membrane of this example is the endosome. And so now it's stuck in the membrane. You've got this Rab stuck in the membrane. And then you've got these effector proteins. And then you also have this other thing going on where you have these kinases that are phosphorylating other things. And basically the whole point is you get this more recruitment of lots of Rab proteins and these tethering proteins in this patch of this membrane. And just think of it as if you've got Velcro, right? Let's say you have some Velcro. If there's one little plastic thing, you think you're going to catch anything? But if you've got like a whole big thing of plastic things, like a normal Velcro that's on your shoe or wherever, will it bind to anything? Yes, of course. But if you got like one little thing hanging out there, is it going to be useful? No. So it's basically kind of making this patch that your vesicles are going to associate with and help with the fusion. Okay. Do you have snares on this? No snares on this picture. Snares. Okay, so we talked about Rab. Well, a very important thing for membrane fusion is snares. So it's also involved in targeting, but more in membrane fusion. And so there's your V snare that's going to be on your vesicle, and your T snare that's going to be on your target. And as the Rab binds to the tether and brings it close, they're going to interact and twist around with this other snare. And what really happens here is you're excluding water. You're excluding water, right? Because membrane, she told me before she was going to be late. It's okay. She's not breaking any rules. If you guys are ever going to be late, don't just not come to class. Email me, and I'll make sure to leave the door open for you. I just don't want the rudeness of like, hey, I slept late. Okay, so if you've got membrane and you exclude all the water, that's going to help with fusing the membranes together. So it's actually brings them tight together and excludes the water, and then you get fusion. Questions? Okay, so obviously snares are going to have to dissociate, right? Because you get the docking and the fusion, and then you just have these snares stuck in your membrane. And that's kind of a waste of snares, isn't it? So you've got these other proteins, NSF and some other proteins that come along. It requires ATP because it's so tightly wound and helps to dissociate the snares so that they can be used in transport of other things in other vesicles. So this is just more about Rabs and vesicles and fusion, and it's just again showing you've got your donor compartment and your target compartment. So this, let's say, would be your ER, and this might be your Golgi, and you're going to have a vesicle. You're going to have a vesicle with Rab bound to it, and it's going to bud off, and you've got your Rab and your snare, and it's going to dock. Now this is an old figure, so it doesn't have the detail that I just showed you, but I like how it shows your target and your donor and your target, and it's going to help with docking and snare pairing. See, there's a way to remember snare pair, and then bring it close together and fusion, and then your GTP is going to be hydrolyzed. It's going to go off, and it's going to actually your Rab is going to be inactive until it's needed again. So we've got proteins moving out of the ER and vesicles. How long can we talk about vesicles for? Let's see. And this is just to explain that they tend to fuse together. Vesicles bud off the ER and will fuse to each other and then turn into a vesicular tubular cluster and then move that way towards the Golgi. And they actually associate with motor proteins and move along microtubules and go towards the Golgi. Right. So that's what it looks like. So there's your ER, and you've got your COP2 coated vesicles budding. Notice your protein coat goes away right away because that's what it says. And then the vesicles bud to each other and become this vesicular tubular cluster, which interacts with the motor protein on a microtubule, kind of like a train on a track, and goes to the cis Golgi, which is the closest part of the Golgi to the ER, and then fuses with the Golgi. So that's what's going on here. So my question for you guys, because you all did your homework and did the quiz, is what is happening over here? What is going on with this? Why are things going from the Golgi to the ER? I keep talking about how you go ER, Golgi. Yes?
I don't remember all the details, but I remember it had something to do with, there were certain ones that they would, like, come out and then they would have to, like, bring them back into the ER because they're supposed to stay in the ER.
Okay, so something about bringing proteins back to the ER because they're supposed to stay there? Yes.
Something about acidic ingredient or something.
Okay, something about pH. Okay.
I was gonna say, isn't like BIP? Doesn't BIP get put out sometimes and then just come out?
Okay, so BIP is an ER resident protein, so we're getting there, right? ER resident proteins need to stay in the ER because that's where they work and where they live. They live and work together. Isn't it horrible? They never get to go anywhere else. They're always at work. When they do go elsewhere, they get sent back. Okay, so they need to go back. What else? Anything else you guys can tell me about? We have something about pH, which we are going to get to. Okay, so good. So there's this retrieval pathway. So sometimes proteins that are involved with folding or whatever is going on in the ER will either accidentally or be bound and on purpose be end up in the Golgi, but they don't work in the Golgi. They need to go back. What's the point? Like if all of the BIP ends up in the Golgi, then we're just going to have to make more BIP, and then we'll have all this BIP in the Golgi that doesn't do anything in the Golgi. So there's this recycling pathway. Or retrieval pathway. And basically the way it works is there's this KDEL, remember KDEL? KDEL, that's just the amino acid sequence. Sometimes it could actually be KKXX. And that sequence will be the C-terminal of proteins, and it will be recognized by a receptor, the KDEL receptor, and it will bind to those proteins, and it will retrieve them back to the ER. And where pH comes in, well, are you guys wondering if KDEL is bound by the KDEL receptor? How come in the ER they're not just stuck bound to the KDEL receptor in the ER, right? It gets bound by the KDEL receptor once it's out. So let's say it's in the Golgi. There's a KDEL receptor that binds, and then it goes back, and then it's released, right? It's released in the ER. Why isn't it still bound, right? If a protein binds to a receptor, why is it going to be suddenly released? That's where pH comes in, right? So that's what you were talking about, right? So the pH, there's actually a pH gradient where you become a little bit more acidic as you go from ER to Golgi. And do you guys understand at all how so let's just go back for a minute. What are the main things that will affect protein-protein interactions? Let's say you're in the lab and you want to prove that two proteins interact with each other, and you're making buffers to try to dissociate them. What are you going to do with your buffers? What different characteristics are going to affect whether or not two proteins can interact or not? Just think about it for a little bit. Yes?
The shape of their active sites.
Well, shape, okay, so yes. So let's say, so he's saying shape. Yes, shape definitely has a lot to do with whether or not proteins are going to interact. But let's say all you can change, your protein is your protein, all you can change is your buffer. You're going to add different buffers and see if the proteins still bind. What can you do with your buffers that's going to disrupt the interaction? Yes?
Maybe you increase the buffer or something.
Increase what in the buffer? Okay, so pH. pH is one thing. I see a hand back there. Sugar. I don't know about sugar. Right. So that was about keeping proteins in the ER, and that had to do with binding. So I see where you're going, right? Because certain proteins will bind, lectins bind to sugars, but this is more like a general protein-protein interaction, not specific to a type of protein. So general. So pH will affect whether two proteins can interact. If you're trying to keep proteins together and you like make something super acidic or super basic, you're going to disrupt that. I saw your hand.
I was just going to say the chemical properties of the chains of proteins. So if you have like hydrogens binding where there would be hydrogen bonding instead.
Okay. So hydrogen. So that would be pH. One way pH would affect, right? So you've got these. I'm kind of going through this exercise because I don't know that people ever thought about this. I didn't think about it till graduate school, so I'm pushing you guys. The characteristics of amino acids determine whether or not proteins are going to interact, right? Right? And if you throw a bunch of hydrogens in there by doing what to the pH? Decreasing the pH, right? Then that will affect whether or not they're going to interact. So what else? So we talked about pH. What's one other thing?
Salinity.
Salinity. Exactly. Salinity. Salt, right? So if you put 500 millimolar sodium chloride into a solution, is anything still going to interact? No. No, I'll tell you that because that's what I do when I want to prove something is ubiquitinated and not interacting with ubiquitin, right? Because ubiquitination is covalent. Interaction is interaction. So if it can survive 500 millimolar sodium chloride, then it's ubiquitinated. Okay, so those are two big things. Yes.
Addition of methanol.
Oh, alcohols. Oh yeah, I'm sure. Yeah, because that dehydrates.
Yeah.
Yep. What else? So the two big ones are pH and salt. And that's why, like, when you talk about buffers will have different pHs, and you can vary that. And when you talk about, and then they'll also have different levels of salt. So when you're working in a lab, and someone gives you buffer blah, 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 and you just put it on your thing, and magic happens, you should look at what's in it and be like, oh, this is a high salt buffer. What is it doing? Why am I using a high salt buffer versus a low salt buffer? And just things to think about. Anyways, yes.
So temperature factor in that?
Temperature, another huge one. Yeah. You know why? What happens when you heat things? Denatures, right. So that's another. So if you want to really test whether or not something's covalently modified versus just interacting, high salt and heat are a good way to look at it. Yeah. Anything else? Okay, so why am I going through this biochemistry lesson? Because we're talking about things that bind in a slightly acidic pH. Now, not super acidic, slightly acidic. So don't forget, it's not as general as I said. So that's why I didn't say acid or base. I said pH. Shifts in pH. Every interaction has an ideal pH that it works at, right? So in the case of the K-del receptor binding the K-del motif, amino acid motif on a protein, its ideal pH is slightly acidic. So it's going to bind tightly at a slightly, I'm not talking a pH of 4, maybe a pH of like 5 or something. And it'll bind tightly, and when it gets to the neutral pH of the ER, it won't bind tightly anymore, and it will release the protein. Okay? So if you understand why, you'll remember it. So hopefully you all understood why there, and when we talk about this on an exam, because this is a really favorite kind of thing to talk about on an exam, you'll be able to answer the questions. So what's going to happen if there's no K-del? So the protein won't go back to the ER. Where will it go? Will it stay in the Golgi? Does anything really stay in the Golgi? Not much. Yeah, so the default pathway in the secretory pathway is secretion, which is why it's called the secretory pathway. So BIP, if you get rid of the K-del, might be secreted. And I might have just answered a question. So here's your practice problem, which I just partially answered. You're studying a protein, so you guys can spend the next few minutes talking with each other. You're studying a protein that appears to be a member of the HSP70 family of chaperones. You believe it is an ER resident chaperone. You look at it using immunofluorescence using a BIP antibody, which is green, and get the following result. Blue is DNA, DAPI, so that's your nucleus. Red is a stain for membrane. Look at that. That's the border of your cell. And then you get green for your BIP, which is your HSP70 chaperone, and it's there. Okay. Do you think? Well, what do you think about? Where do you think this is? ER. Where else might it be? Might be in the Golgi. So you're not sure, right? You don't know. So that looks nice. Now, if you were really being a good scientist, you wouldn't have done a stain with red for the membrane. You would have done like ER. There are ER specific, like calnexin, right? You can stain calnexin because that lives in the ER. That would be how you know. But so you don't know. And so you see BIP in the ER in Golgi. You're pretty sure. And you decide to look for a retrieval signal. Right? Because that might be, because right now you really don't know what this BIP is. We know because we've learned about it, but you don't know. You're a scientist, you're studying, and you don't know. And you decide to look for a retrieval signal. Do you see a retrieval signal? And how do you prove it's a retrieval signal? What experiment do you do? And what do you expect to see? So stop, chat with your friends, and we'll reconvene in a minute or two.
Right, yeah. Prove it. She said, send it out. Yeah, don't be with the myself. At the end, there's
Which is why they're in the PowerPoints, because you'll go over them, and when you're asking me for your review sheet, what you'll do is you'll go through all this, pull all these out, copy and paste them into a document, and you'll have a whole list of questions to go through, which hopefully someone will remember the answer to.
What do you say in the thing?
Yeah, it's true.
Is this retrieval pathway also how they keep the gold from getting too big in the air from getting too small? Because the best transport.
Because of the membrane? Is it because you mean because there's all this addition?
Yeah, because like the ER is just like donating.
Right. So ER has to, I think membrane is made in the ER. There are actually enzymes that are involved in making more membrane there. But don't forget, right? So it's a two-way street, exactly. So you've got ER, Golgi, plasma membrane, right? Because you also don't want your cell to get huge, right? Because things keep fusing. Things keep fusing with the plasma membrane. How does it not get totally huge? There's a whole other process, right? Endocytosis, where things butt in. And so it kind of maintains this balance. And I think ER will get bigger in different kinds of cells. Let's say you're making tons and tons of protein as part of, I don't know, an immune response or something. Things might get bigger for a little while. Okay, guys, in the interest of time, do you guys see a retrieval signal? Hopefully everyone caught that because it's like screaming red. I may as well have highlighted it red because it's the last four. And then sometimes you'll just have KK, XXX, meaning any amino acid. And you can see actually there's a fair amount towards the end. There's also KK here. I think there's another KK here. So when you start to have a lot of that towards the end, so it's not just KDEL. KDEL is like your typical kind of thing. But you'll see a lot of it there, and that's really going to mediate that interaction with the receptor. Okay, so how do you prove it? So you see K-del. Do you just say, well, I see K-del? So if you were publishing this paper, the reviewer would say, well, prove it. Show me that it's a retrieval signal. What would you do?
Read the sequence and then track it.
Okay, so remove K-del and see where it goes. Right, and where would it go? Where? Where? Someone raise your hand. I hear a lot of people mumbling. Yes.
The Golgi towards the membrane.
Out of the Golgi towards the membrane, probably secreted. Right. So you could check your supernatant and see if it's secreted out. Because should BIP be outside the cell? No. Yes.
Could you actually use like you were talking about buffer? Could you actually like compare the acidity?
So it's hard to do inside a cell. So when you're looking at where things are in a cell, you need to keep a cell intact. Because once you disrupt a cell, then you don't know where things are in the cell, right? Like once you break it open, things spill out and go everywhere. So the buffer, so he said, can you mess around with buffers and pH and salinity and that kind of stuff? And there might be a way, but let me just think real quick. It's just hard because you can't inject buffer into the ER and change the pH of the ER. Well, maybe you can. You can with the lysosome. You can with the lysosome. So maybe, okay, so lysosome is highly acidic, and chloroquine, I believe, raises the pH. Who knows what chloroquine is? This is from a malaria endemic area. Okay, thank you. Chloroquine, the way it works, I believe, is it raises the pH of the lysosome. Okay, so let's move forward with what's your name again? Fritz. So can you change the pH of the ER or the Golgi and see what happens? So that's kind of the direction you were going in, right? And so let's say there was a drug that you could give a cell that would change the pH. That would answer something, but not directly. It wouldn't directly say, is K-del a retrieval signal? It might say that pH is required or not and it might be a nice experiment to do alongside with deletion to support it even more, what you're trying to say. Yes?
Could you also stain both the and the receptor and see if you get like protein?
Yes, so so usually so the comment was, can you just stain the receptor and the protein and see if you get colocalization? Well, yes, you can, but again, the K-del receptor is going to be everywhere, right? Because it's in the membrane and it's going to be retrieved. But you can, to change that, you can stain your protein, and I guess what you always need to do is chop off your K-del. If you want to prove a sequence does something, you need to remove it and show it doesn't do something. But co-staining is a very common way to show that things interact. And you can show they interact. You can like, usually when you want to prove a point for a paper, you need to show something in 10 different ways. So you would chop it off, see where it goes. You would change the pH, see that it doesn't work. You would also do co-staining and see that things interact. And you would do it all together, and then the reviewers who are looking at your paper would say, okay, she showed this, or he showed this in 10 different ways. It must be true. Okay, you guys can go because now we're kind of late.